Our bodies are in constant motion. Whether we are walking, talking, or even breathing, our muscles are hard at work. Muscles power our every move, from the simplest blink to the most complex athletic feat. But have you ever stopped to wonder how these incredible tissues actually work? Understanding muscle physiology is for everyone who wants to appreciate the marvels of the human body. By delving into muscle contraction and relaxation, we gain a deeper understanding of how our bodies move and why exercise is essential. Just like an orchestra relies on the coordinated efforts of its musicians, our muscles depend on the intricate interplay of various components to function correctly. Let's uncover the science behind muscle contraction and relaxation, revealing the astonishing mechanisms that empower us to move, breathe, and live our lives to the fullest. To truly grasp the complexity of muscle function, we must first journey into the microscopic world within our muscles. Imagine a telescopic view, zooming in on a single muscle. You would see that it's not a single uniform structure but rather a bundle of smaller units called fascicles. These fascicles are like miniature cables within the larger muscle cable, all working together to generate force. Each fascicle is further divided into numerous muscle fibers, which are the individual cells of muscle tissue. These fibers are long cylindrical cells that contain multiple nuclei. Think of them as the individual threads within our muscle cables. Within these muscle fibers lie the true secrets of muscle contraction, the myofibrils. Myofibrils are thread-like structures that run the length of the muscle fiber. They are composed of repeating units called sarcomeres, which are the basic functional units of muscle contraction. At the heart of every muscle contraction lies a fascinating interaction between two protein filaments, actin and myosin. These filaments are arranged in an overlapping pattern within the sarcomere, creating the striated appearance characteristic of skeletal muscle. Actin, the thinner filament, forms the backbone of the sarcomere. Myosin, the thicker filament, has numerous heads that protrude outward resembling tiny oars. These myosin heads are the force generators of muscle contraction. They have binding sites for actin and ATP, the energy currency of the cell. When the muscle receives a signal to contract, the myosin heads attach to the actin filaments, forming cross bridges. These cross bridges then pivot, pulling the actin filaments closer together. This pulling action, repeated across countless sarcomeres within a muscle fiber, causes the entire muscle to shorten, resulting in contraction. The coordinated action of millions of these tiny handshakes produces the incredible force that allows us to move our bodies with precision and power. Now that we've met the key players, let's explore how they interact to produce muscle contraction. This intricate process is elegantly explained by the sliding filament theory, a cornerstone of muscle physiology. Imagine a rowing team propelling a boat across a lake. The oars, representing the myosin heads, grip the water, which represents the actin filaments. As the rowers pull on their oars, the boat moves forward. Similarly, in muscle contraction, the myosin heads grip the actin filaments and pull them inward, causing the sarcomere to shorten. This process of myosin heads attaching, pivoting, and detaching from actin filaments is cyclical and requires energy. This fuel is provided by ATP, the energy currency of the cell. As we've learned, muscle contraction is an active process that requires energy. This energy is supplied by adenosine triphosphate, commonly known as ATP. Think of ATP as the battery that powers muscle contraction. Without ATP, the myosin heads wouldn't be able to detach from actin, and the muscle would remain in a contracted state, a condition known as rigor mortis. ATP binds to the myosin head, providing the energy needed for the myosin head to detach from actin and become recocked, ready for another cycle of contraction. The breakdown of ATP into ADP, adenosine diphosphate, and inorganic phosphate releases energy that powers the myosin head's pivoting motion, pulling the actin filament. This process of ATP breakdown and regeneration is continuous, ensuring a constant supply of energy for muscle contraction. This highlights the importance of a balanced diet and proper respiration in providing our bodies with the energy they need for everyday activities and physical performance. While ATP provides the energy for muscle contraction, calcium ions, Ca2+, act as the crucial signal that initiates the process. Think of calcium as the conductor of an orchestra, signaling the musicians to begin playing. In a relaxed muscle, calcium ions are stored within the sarcoplasmic reticulum, a specialized network of membranes within the muscle fiber. When a nerve impulse signals a muscle to contract, calcium ions are released from the sarcoplasmic reticulum into the cytoplasm surrounding the actin and myosin filaments. This surge in calcium ions is the trigger for muscle contraction. Calcium binds to a protein called troponin, which is attached to the actin filament. This binding causes a conformational change in troponin, which in turn moves another protein called tropomyosin. When calcium binds to troponin and shifts tropomyosin, these binding sites are exposed, allowing the myosin heads to attach to actin and initiate the sliding filament mechanism. So far we've explored the intricate dance of proteins and ions within the muscle fiber. But what initiates this complex sequence of events? The answer lies in the nervous system, the body's control center. The nervous system acts as the maestro, sending electrical signals that instruct our muscles to contract or relax. When you decide to move, your brain sends electrical impulses down your spinal cord and through motor neurons, which are specialized nerve cells that connect to muscle fibers. Each motor neuron can innervate multiple muscle fibers, forming a motor unit. When the nerve impulse reaches the neuromuscular junction, the point where the motor neuron meets the muscle fiber, it triggers the release of acetylcholine, ACA, a neurotransmitter. ACA binds to receptors on the muscle fiber membrane, initiating a cascade of events that leads to muscle contraction. Not all muscle contractions are created equal. The two main categories are isotonic and isometric contractions. Understanding the difference between these contractions can help us better understand how our muscles function during various activities. Isotonic contractions involve a change in muscle length while the tension remains relatively constant. Imagine lifting a dumbbell. As you curl the weight upward, your bicep muscle shortens, performing an isotonic contraction. There are two types of isotonic contractions, concentric and eccentric. Concentric contractions occur when the muscle shortens as it contracts, such as when lifting a weight. Eccentric contractions, on the other hand, occur when the muscle lengthens as it contracts, such as when lowering a weight slowly and with control. Isometric contractions, in contrast to isotonic contractions, occur when the muscle develops tension without changing length. Think of pushing against a wall. Your muscles are working, but there's no movement in your joints. Just as there are different types of muscle contractions, there are also different types of muscle fibers, each specialized for specific types of activity. The two main types are slow twitch, type 1, and fast twitch, type 2 fibers. Understanding the characteristics of these fiber types can help tailor exercise programs to specific goals. Slow twitch fibers, as their name suggests, contract slowly and are resistant to fatigue. They are rich in mitochondria, the powerhouses of the cell, and rely primarily on aerobic respiration for energy production. Marathon runners, cyclists, and swimmers tend to have a higher proportion of slow twitch fibers. Fast twitch fibers, on the other hand, contract rapidly and generate more force than slow twitch fibers. They fatigue more quickly and rely more on anaerobic respiration for energy. Sprinters, weightlifters, and power athletes typically have a higher proportion of fast twitch fibers. Most muscles contain a mixture of both slow and fast twitch fibers, but the proportion varies depending on genetics and training. One of the most remarkable aspects of muscle tissue is its adaptability. Our muscles are constantly remodeling themselves in response to the demands we place upon them. This phenomenon, known as muscle adaptation, is the foundation of strength training and physical conditioning. When we engage in regular exercise, especially resistance training, we create microscopic tears in our muscle fibers. In response to this damage, our bodies repair and rebuild the muscle tissue stronger than before. This process of muscle repair and growth, known as muscle protein synthesis, is driven by a complex interplay of hormones, growth factors, and nutrients. By consistently challenging our muscles with resistance training, we stimulate this process, leading to increased muscle mass and strength. Whether you're a seasoned athlete or just beginning your fitness journey, understanding the principles of muscle adaptation can help you optimize your training and achieve your fitness goals. From the microscopic dance of protein filaments to the coordinated efforts of the nervous system, muscle contraction and relaxation are a testament to the elegance and efficiency of the human body. Understanding the science behind muscle function is not just about satisfying our curiosity, it's about empowering us to take control of our physical well-being. By appreciating the adaptability of muscle tissue, we can unlock our potential for strength, endurance, and overall health. So, the next time you marvel at an athlete's performance or simply appreciate the ease with which you move through your day, remember the intricate symphony of muscle magic at play. Within each muscle fiber lies a world of wonder, a testament to the remarkable capabilities of the human body.